Hello all and welcome. I would like to present you to BioStrand. BioStrand is a Belgian-based bioinformatics company, and we have made it our mission, and we're passionate about building the next generation technology that makes omics data truly effective. Well, making omics data effective, where does it come from? First, omics data is larger, bigger than astronomy data. Next to that, omics data is extremely heterogeneous. We're talking about sequence data, text data, compound data, which relates to chemical molecules, metadata, clinical outcome documents, electronic patient records, articles, a whole sphere that comprises omics data. But next to the fact that it's huge, that the fact that it's very heterogeneous and siloed, the biggest problem of all is that we can just analyze with current techniques a small fraction of this pool of omics data. It's not integrated at all, and that leads to the fact that, let's say, substantial knowledge today still remains untapped. To solve this problem, basically, we came up with a way to encode all this omics data, all this vast pool of heterogeneous data. With our way of encoding, we basically can integrate all the silos, normalize all the data into a central repository, and from this repository, it's very easy to also manage all the different and dispersed data sets. But most of all, the best thing of all is that from this one single model, from this one single framework, you're capable of analyzing everything that there is to it. To give you some more insight on how we deal with all this data, I would like to give you a short overview of our platform. On the left, well, as omics data is extremely diverse and can come from a multitude of sources, we are capable of encoding virtually any source. Whether it's data coming from the public domain or your own proprietary databases, it can all be encoded and integrated using our proprietary HIFT patterns. Next to historic data, new data is produced each minute. Indifferent of what system is generating this data, our HIF patterns can encode and ingest it. In the same way, we can also add textual sources, like electronic health records, clinical trial records, and lab records. From encoding all these diverse data sources, we can integrate them into one single data framework, and from this framework, any type of analysis is possible, being on all the data, all the relevant data. So this leads to petabytes being analyzable in near real time. And that can range from a simple search across databases to a cross-database comparison, but also more comprehensive tasks that leverage the latest and greatest AI and machine learning algorithms benefit greatly from this hyperscalable and multidimensional data framework. A nice side fact, well, our platform is fully agnostic. It can be hosted anywhere from cloud environments to on-premise deployments. Within our platform, one of the key components is the Intersystems IRIS Health database and the I know NLP technology that help us to build our central repository. Now, to give you an example on how we leverage these building blocks, I'd like to run you through a few examples. First, I'll provide an example where we make all the data from the public domain actionable. Second, there is an example where we leverage bottom-up knowledge extraction to immediately interpret and discover novelties in experimental data. And third, an example of the power and value of having such a hyperscalable multi-omics platform. Making public data actionable, that's where we have our first web tool, Retrieve and Relate. With a simple click of the button, you launch a query against 500 million of sequences. Next to the fact that it just takes seconds to find results, the big difference is that it is launched across all the different data sets, 14 in total, around 500 million sequences. And once you retrieve your results, you can start to play around with the relevant dimensions associated to your sequence data. So leading to insights in just seconds. As an example of fully integrated data to enable bottom-up knowledge extraction, I'd like to show you our pathway enrichment analysis process. It is an extremely cumbersome task today. In such analysis, you try to find associations between tens of thousands of genes, which you first have to label. That in its own is an extremely time-consuming task. Then, based on this pool of genes, you try to make sense, pointing towards literature sources and other unstructured data sources to define their associations. Currently, that takes around 20 consecutive steps, and if you're lucky, you can do it in half a day. Still then, the end result is a cloud of single words, and in order to really make sense of them, let's say, discover the relevant literature, that's a manual and additional process that has to be taken. In our way of analysis, in one step, we can go from raw data to insight in just a few minutes. We don't just provide a word cloud of associated terms, but we build a complete knowledge graph. Leveraging I know, we can retrieve related concepts that don't even have to be in close proximity in the text. But without pre-training, we can build ground-up knowledge graphs that can also point to the relevant literature sources. Result is that anybody can start to make sense of experiments, uploading raw data, and as a result, you have a full semantic graph that points to the relevant literature sources and really helps you to make sense of the pathways of the associations between your genes. This in part helps them to really define the process within their drug development. For our third example, I'd like to run you through our analysis of 1 million SARS-CoV-2 sequences. Very unique because currently techniques can only analyze a few 10,000. So as a starting point, we compared SARS sequences, those million, by collapsing them, and this helps us then to find commonalities and variations, as can be seen on the graph in the center of the screen. This on its own is very useful because it tells you, okay, where are the variations, where are the commonalities, but it does not tell us anything about what we are seeing. And this is where we really differentiate ourselves with the rest of the technologies out there. As we can integrate any related data source, we can use this multidimensional model to immediately make sense of these commonalities and variations. Take into account variations, geographical data, taxonomy data, we can point to the meaningful parts within these sequences. Not only limited to direct associations, we can also integrate data from electronic health records and trial data to assess the impact of, for example, the variations more carefully. As our model is not static and viruses constantly mutate, we can easily add them to the data model one by one, without having to build from scratch the whole model again. From our specific SARS-CoV-2 viruses, we can easily do cross database comparisons. This helps to identify overlap with known drugs, to repurpose them, or to find epitopes, which are, let's say, the parts where a vaccine can be targeted to. As a result, we can continuously update the data model, spot the locations where we need to adapt, for instance, vaccines in future, because as time moves on, clinical phenotypes can also be, let's say, differentiated and found. So, in summary, the situation today is that we have a lot of unstructured data that resides in separated silos and is extremely expensive to integrate, to work with. It takes weeks rather than days and minutes. And in the end, we can say that all this data we have remains a sunk cost because it's not a true asset. With a technology like BioStrand, we can really see the added benefit of structuring it into one single, by default, integrated data model to make all data actionable in just minutes. This, in part, leads that all the data, all your omics data, is an asset. And omics data truly is an asset and should be a core asset. Multiple industries produce or rely on omics data today, but they are still separated and it remains difficult to connect this knowledge. Supporting these diverse industries by integrating their data, in the end, we can support the patients with faster, more tailored drug development, which in its part progresses healthcare systems. Thank you very much for your attention. And in case you have questions, I invite you to contact either myself, Arnout, or my colleague Audrey. Please feel free to have a look on our website and I hope to talk with you soon. Thank you.